Discover how the unique structure of mitochondria enables efficient ATP production during cellular respiration. The mitochondrion is the powerhouse of the cell, with a specialized structure perfectly designed for energy production. Mitochondria have a unique double membrane structure that creates distinct compartments. The outer and inner membranes create separate compartments that allow different chemical reactions to occur simultaneously. The outer membrane is smooth and permeable, while the inner membrane is highly folded into structures called cristae. This double membrane system creates compartmentalization essential for the electron transport chain. The inner membrane folds dramatically to create cristae, massively increasing surface area. The folded cristae can increase the inner membrane surface area up to 5 times compared to a smooth membrane. Compare a smooth membrane with a folded one. The folds dramatically increase the available surface area for ATP production machinery. More surface area means more space for ATP synthase enzymes, which directly translates to more ATP production. The space between the two membranes plays a crucial role in creating the proton gradient. Protons are pumped into the intermembrane space, creating a concentration gradient that stores potential energy. Protons marked as H+ accumulate in the intermembrane space, creating a high concentration that drives ATP synthesis. High proton concentration equals stored potential energy, just like water behind a dam. The innermost compartment, called the matrix, is where the citric acid cycle occurs. The matrix contains all the enzymes needed for the citric acid cycle, also known as the Krebs cycle. The matrix is packed with enzymes that break down pyruvate and generate electron carriers, NADH and FADH2. In the matrix, the citric acid cycle produces NADH and FADH2, which carry electrons to the electron transport chain. ATP synthase is the molecular machine embedded in the inner membrane that actually produces ATP. ATP synthase harnesses the flow of protons down their gradient to mechanically synthesize ATP molecules. Protons flow through ATP synthase like water through a turbine, spinning a rotor that catalyzes the conversion of ADP to ATP. The enzyme adds a phosphate group to ADP using energy from the proton gradient to create ATP. The electron transport chain consists of protein complexes embedded in the inner membrane. 4 large protein complexes work together to transfer electrons from NADH and FADH2 while pumping protons across the membrane. Electrons flow through complexes 1 through 4, with complexes 1, 3, and 4 pumping protons to create the gradient. Electrons from NADH flow through the complexes, releasing energy used to pump protons against their gradient. Now let's see how all these structural features work together to produce ATP efficiently. The complete system shows cristae providing surface area, complexes pumping protons, and ATP synthase using the gradient to produce ATP. The mitochondrial structure enables efficient ATP production through several key mechanisms. First, the large surface area from cristae allows many electron transport chains to operate simultaneously. Second, compartmentalization creates the space needed to build up a strong proton gradient. Third, high enzyme concentration in the matrix ensures rapid processing of substrates. Fourth, many ATP synthase molecules work in parallel to maximize ATP production. The mitochondrial structure represents an elegant solution to the challenge of efficient energy production. From one glucose molecule, the mitochondrial system can produce 32 to 34 ATP molecules, representing about 40% efficiency. One glucose molecule yields 32 to 34 ATP through the combined processes of glycolysis, citric acid cycle, and electron transport chain. Every structural feature of the mitochondrion contributes to maximizing ATP yield from each glucose molecule. Let's review how mitochondrial structure enables efficient ATP production. The double membrane system creates separate compartments essential for the proton gradient. Cristae dramatically increase surface area, allowing more electron transport chains and ATP synthase molecules. The matrix concentrates all enzymes needed for the citric acid cycle in one location. The intermembrane space acts as a reservoir for the proton gradient that powers ATP synthesis. In conclusion, the mitochondrial structure is a perfect example of how biological form enables function in cellular energy production.